Hello, Honors Biology. This is Mrs. Kozian. This is Unit 3 Cells, and this is going to be video 3A, Passive Transport. Okay, we're going to talk just in this first video from lipid bilayer all the way through this term called facilitated diffusion. So this is Unit 3 Cells, video 3A, Passive Transport. Here is our standard that we're trying to meet in this portion of the unit. We are going to explain the role of the cell membrane as a highly selective barrier. Another word is semi-permeable. We're going to talk about the process of diffusion, osmosis, or passive transport. Then we're going to talk about active transport. First term for you to know, lipid bilayer. Now I know that we talked about this in the first unit. We called it a phospholipid bilayer. Okay? Remember, lipids are fats, and bi means two layers. It's just another word for your cell membrane. Now it's composed of two layers of lipids that are, this is the flexible barrier for our cell. Now this is a one-dimensional cross-section of it. Water exists on both the outside and insides of our cells. You guys can see the glycerol head here and then the fatty acid tail. Okay. It's very important that, because we know that water is a polar molecule, these hydrophilic heads are nonpolar. And remember, nonpolar and polar, they won't mix together. So you can think of the hydrophilic heads almost like the oil part of the lipid, and then water is polar, so they won't mix together. So this ends up being sort of waterproof. Okay. Now this picture, it's more of a three-dimensional model of the phospholipid bilayer. You guys can see this is very similar to the one-dimensional model you saw in the previous slide. But the phospholipid bilayer covers the entire outside of your cell. Oh, that's my cat meowing at me. All right, I kicked the cat out. So your phospholipid bilayer, it acts three-dimensionally around the outside of our cell because our cells are three-dimensional. So the outside, you can see the glycerol heads here, they fit together almost like scales. But how do things get in and out of our cell? That's what we're going to talk about in this portion of the unit. We're going to talk about the cell membrane as a selectively permeable membrane. Okay, now the first thing that we're going to talk about, you guys, it's called passive transport. Passive means you don't need any energy, and diffusion is an example of that. So here's my example of diffusion before you even start writing down definitions. If I stand in the corner of the classroom and I spray air freshener, okay, let's say we have a lovely flower-smelling air freshener, and I'm way in the corner by the door. What eventually happens to the scent? At first, the people who are really close to me smell it and it's very strong. People at the other end might not smell that air freshener. But as it diffuses or moves and reaches what we call equilibrium, everybody in the room would smell it equally. It's called passive transport. It doesn't need any extra energy. The first example of that is diffusion. Molecules of any type of solute, remember solute is a solid dissolved in a solvent, like air or water, moving from a high concentration to low concentration until it reaches equilibrium. So you could think of this as a container with your solvent might be water. Your solute might be something like sugar. Okay? You have a semi-permeable membrane here. And your solute is going to diffuse across that membrane. Material is going to diffuse across that membrane until it reaches equilibrium. Now the thing to remember is, once we've reached equilibrium, we don't stop. Particles might go back and forth across that membrane, but the particles don't just stop moving when they've reached equilibrium. Okay, and here's our definition of equilibrium. The concentration is going to be equal in all parts of the solution. Take a peek at these three. Okay, you can see over time, as things start to move across the cell membrane, or the solute moves across the cell membrane, eventually it reaches equilibrium here. This picture is at equilibrium. Now, concentration gradient. Concentration gradient is where you have a difference in balance of concentration in the solution. So that means you have one part or portion that has more solute than another. Think of it like a hill. The higher the concentration gradient, or the bigger the difference in your concentration, the steeper the hill. So we have a very large concentration gradient here. We have a huge difference between the concentration on this side of the solution and this side. So there will be a lot of diffusion, a lot of passive transport. And then here we have a much shallower or not as steep our concentration gradient. And here there's almost no concentration gradient. There's no difference. You're at equilibrium. And here's another picture just to show you concentration gradient. We have high concentration to low concentration, like moving down a hill. Osmosis. Osmosis is an example of passive transport that only moves water. Osmosis is diffusion of water, doesn't require energy. Now, up until now, we've been saying we go from high concentration to low. So when you look at this first picture, you may think, well, I have a higher concentration of solute here than here. Why don't I just move some of those over? Well, the problem with that is when you have a membrane that is very selectively permeable and your particles are too large to get through it, what we have to move instead is water. So we change the concentration of the whole solution. So here we have like a much more diluted solution. Here we have a much more concentrated solution. So what I'm going to do is I'm going to move water from high concentration to low concentration of water until the overall concentration is balanced. So you can see here I ended up with more water, but, oops, sorry. I ended up with more water, but now my concentrations are equal between the two. 3 words to describe osmosis are isotonic, hypotonic, and hypertonic. The first one's called isotonic. That's when we have an equal concentration both inside and outside the cell. This thing right here is supposed to be the cell membrane, this is outside of the cell, and this is the inside of the cell. Isotonic, everything's equal. Hypertonic. A hypertonic solution would be one that we would put a cell in that has a way higher concentration of solute. And I defined it here, I said higher concentration outside the cell. That's just because that's what you see in this picture. It doesn't always have to be outside the cell. Hypertonic just means you have a really high concentration of solute. And what that's going to do is it's going to take water out of the cell. Remember, water in osmosis moves from high concentration to low concentration. So the water is actually going to be pulled or kind of sucked right out of this cell. So this cell is probably going to dry out. If this cell is, let's say a normal human skin cell and this were salt water, and your skin gets dry in salt water, it's because the actual salt water is concentrated like this, and our cell is like this. It's pulling water out of our cells. Hypotonic is the last one. Okay, hypotonic is where you have a lower concentration of solute outside. So I have really high concentration of water, low concentration of water. Remember, we want to go from high concentration to low concentration. So the water is going to move into the cell. And this is where if we have the concentration is so different, it can actually pull too much water into the cell, and the cell can explode. And I always say hypotonic, it's po makes me think of poor. Okay, like this is very poor in solute. High in water, poor in solute. Just some pictures. A hypotonic solution, a living cell. The water goes in, remember high concentration to low, and the cell can get kind of waterlogged. Isotonic, everybody's happy, pretty normal. Hypertonic is going to take the water out of the cell. Last piece of passive transport here, guys, facilitated diffusion. Think of it like a lock and key. You don't need extra energy for a lock and key. It doesn't, plug in. You don't need to have any energy there. We use energy to turn it, but think facilitated diffusion. It's like a lock and key. It's not plugged in. It doesn't use electricity. It's going to move specific molecules across the cell membrane through protein channels. So the specific molecule comes in, unlocks the lock, and it opens on the other side. High concentration to low concentration. Passive transport again, remember that your molecules never really stop moving. They'll reach equilibrium, but they'll constantly sort of move up and down around it. This is a graph kind of showing the rate of transport. Facilitated diffusion can go through transport quite a bit faster than simple diffusion because we're using those specific protein channels. And that was it, you guys, for Unit 3, Video 3A, Passive Transport.